You're listening to Fire Ecology Chats, a podcast series by the Association for Fire Ecology.
Hello, everyone. My name is Bob Keane. I'm the editor-in-chief of the internationally renowned journal Fire Ecology. Everybody wants to publish in Fire Ecology now because we're getting such a huge impact factor, and I'm glad that you came to hear about one of the papers that has really driven up our impact factor. The paper we're going to be talking about today is Towards Predicting Flammability of Sierra Nevada Mixed Conifer Forests. Drought stress and fuel moisture are strongly linked in angiosperms, but decoupled in gymnosperms. Isn't that interesting? And today we have three of the authors here to talk about that paper: Indra Boving, Joe Celebrezze, and Leander Anderegg. Could you introduce yourself, guys? How about Indra? Do you want to go first?
Sure thing. My name's Indra Boving. I'm a PhD candidate at the University of California, Santa Barbara.
I'm Joe Celebrezze. I'm also a PhD student here at UC Santa Barbara.
Hi, I'm Leander Anderegg. I'm an associate professor at UC Santa Barbara.
Well, I want to thank you all three for coming today and talking about this really interesting paper. I really enjoyed reading, and I'm sure the readers will too. Maybe you want to tell them a little about the paper. Indra, you want to talk about a quick and dirty recap of what the paper is?
So very broadly, we know that wildfire risk and drought stress are pretty tightly linked, but something we don't understand as well are the exact mechanisms through which drought can change flammability, especially in living plants. So in our study, we focused specifically on plant hydration, so the amount of water in living plant tissues and how it directly affects tissue level flammability. We focused on this by measuring both hydration and tissue flammability in a lab setting. And then a second important unknown for how we link drought stress and fire behavior actually has to do with how different disciplines characterize plant hydration. So the way we measure plant hydration differs between fire scientists and plant physiology, where fire scientists tend to focus on this metric termed live fuel moisture, which we also call LFM. LFM describes basically the ratio of water to dry matter in a plant sample, while studies on drought stress tend to focus more on water potential, which is this pressure-based metric of water stress in plants. So to kind of address these missing links between these different disciplines, we characterize the relationships between LFM and water potential in both lab and in field settings and across different species up in the Sierra Nevada.
Yeah, I really enjoyed the paper because of the link. A lot of people don't think ecophysiology has anything to do with fire. And I will tell you what, it is all about ecophysiology and drought stress and live fuel moisture content and so on. Can you tell us why this study is important, and especially in review of the fact that should we use live fuel moisture as a fire hazard risk rating or what?
I couldn't agree more, Bob, that ecophysiology is really important here. And I think that's because live fuel moisture is a really great monitoring tool. I think we would all agree that. And it is foundational in a lot of fire risk assessment. But the key is that there are a whole bunch of ways that drought can actually influence fire risk through altering live fuel moisture or the amount of water in plant tissues. And at the end of the day, what we'd like to be able to do is predict fire risk mechanistically rather than just monitor it through our continuous monitoring of live fuel moisture. So if we want to, for instance, use mechanistic vegetation models to predict live fuel moisture and fire risk in future climates, or maybe in contrasting management scenarios, we actually have to understand the link between drought stress in vegetation and the water that's in vegetation, that live fuel moisture. So it really comes down to digging into those mechanisms so that we can predict rather than just explain post hoc or use empirical relationships.
Yeah, I hear you. Is having a fire danger indice that you cannot predict into the future, right? I mean, and mechanistic models will be able to do this. So, Joe, why don't you tell us a little more about your methods that you used in this study?
Yeah, of course. So Indra and I went out to the Sierra Nevadas in California every couple of weeks during the summer of 2020, and we measured both drought stress and live fuel moisture for a bunch of different individuals across 6 dominant species in that area. More specifically, we looked at 3 conifer species and 3 angiosperm species. As we revisited the same individuals throughout the summer, we also looked at their phenology. So using the angiosperm species, for example, we looked at things like when their leaves were fully expanded, when flowers were present, and when they were fruiting. After about 5 or 6 times out in the field in the late summer, when the plants were rather dry and the fire risk was rather high, we collected a whole bunch of branch samples, put them in the back of our truck, and brought them back to the lab for further testing, including flammability tests. When we got back to the lab and prepared our samples for testing in the flammability chamber, it was actually confirmed to us about how high the fire risk was in that area. The creek fire began and started to burn very near to our field site. By the time that was done burning, it ended up being one of the largest fires in California history. Anyways, as the creek fire scorched the forest, Indra and I were in the lab scorching little segments of plants in our flammability chamber. So we allowed these branches to dry on the laboratory bench tops, and as they dried, we tested their drought stress, live fuel moisture, and their flammability. And I could go on for 10 to 15 minutes talking about different flammability chambers and how we quantified flammability, but I'll try to hold myself back in the interest of time. So in brief, we placed our samples atop a wire mesh suspended above a hot plate in a preheated chamber. A Bunsen burner located above the sample acted as an ignition source. And then, using video recordings of these flammability tests, we took various measurements of things such as flame height, time to ignition, and flame duration, which represented different components of flammability. And then, of course, we ended up analyzing that data that we collected.
Yeah, it was really interesting how you did that. Is there anything more important to show that the importance of your study is the fact that there was a fire brewing in the area? That's great. Indra, well, how about a peek at the results?
Sure. So broadly, we found that the angiosperms in our study showed pretty consistent relationships between water potential and live fuel moisture and those metrics and flammability, while the gymnosperms are actually much less predictable. It seems like that inconsistency might stem from how these different groups of plants manage their water during drought or how they shift their tissue structures, especially as seasonal phenological patterns occur. Somewhat intuitively, we also showed that as the plants lost water, their tissues tended to become more flammable. Specifically, they tended to ignite sooner. And then the last main finding, which is maybe more surprising, is that we found that live fuel moisture is generally a better predictor of flammability than water potential. And this was true even for the angiosperms, which had those more consistent relationships. Again, it seems like the kind of dry matter is the culprit here. LFM captures both plant hydration and the buildup of dry matter, again, during drought or as seasonal leaf development happens.
Yeah, I thought that was important too. The fact that water potential did not predict as good as fuel moisture content, that was pretty interesting.
Mm-hmm.
So, Leander, can you relate how we predict and study drought and fire risk in the future?
Absolutely. So I think the good news from our findings were that the link between drought and live fuel moisture is pretty direct in angiosperms. And that's great. That means that the difference in live fuel moisture as it evolves over the course of a season or across the landscape is more directly related to how wet versus dry those plants are. And that we can study that in the lab by using these lab dry down techniques that are by far and away the most easy way to try and link water potential and live fuel moisture and flammability. The bad news is that for conifers, the live fuel moisture dynamics that we saw in the field are not strongly linked to how wet or dry the trees are, which means that one, I think we can't study the drought live fuel moisture links in conifers in lab dry downs. That's a bit of a bummer. It also means that the dynamics of live fuel moisture in conifer systems through space and time are probably going to be more of a question of how drought speeds up or slows down canopy development and canopy phenology. So it's actually a phenology question, as well as an allocation question, how drought changes the relative proportion of fresh, wet tissues versus old, dry tissues. And that those are areas that will be important to dig into in order to understand how drought drives flammability in conifer systems.
Really interesting. Thanks. So, Joe, you get the standard question that I ask all the people that come on the podcast is, what are the implications of the study? How do these results implicate how we're going to manage fire risk and drought in the future?
Yeah, so building upon what Lee and Indra have already pointed to, we saw that there were key differences in how species and functional groups both respond to drought and their seasonal LFM dynamics, and then the implications on flammability. So we saw that although live fuel moisture and flammability consistently have a relationship where drier fuels lead to more flammable plants, there were key differences in the thresholds in LFM that lead to more flammable plant matter. So these observations indicated to us that we need to expand live fuel moisture monitoring across different species in different communities. And by increasing these monitoring efforts and also conducting more of these laboratory flammability tests, we could work towards more nuanced wildfire risk assessments by accounting for these species or functional differences.
Indra, you get the last question. Do you recognize any co-authors or funding agencies?
Yeah, awesome. Thanks for the opportunity to do this. I guess first off, we'd like to acknowledge the traditional custodians and owners of California and recognize their continuing connection to the land upon which our research was conducted. That's particularly the Chumash, Mono, and Wenatchee tribes. And then we'd like to recognize Max Moritz, who's our co-author who couldn't join us today, and also Anne-Marie Parkinson, who led some of our fieldwork over the course of those 2 summers. And then for funding, the UC National Laboratory Fees Grant funded much of this work, as well as the CAL FIRE Forest Health Research Grant.
Thank you, all 3 of you, for a wonderful talk today. I really enjoyed this paper, and I love how you bridged ecophysiology with fire and fire management. It's a wonderful paper, and if you listeners want to read it, guess what? Indra and the crew have already paid the freight for you to go ahead and download that paper, and you can download it from our website for free and read all about this wonderful study. Thank you again, you 3, for joining us.
Thank you for
Thank
having
you, Bob.
us.
Yeah, thanks so much for having us.
And thank you, listeners, for listening. And please tune into the next Fire Ecology Chat.
Thank you for listening to Fire Ecology Chats, a podcast series by the Association for Fire Ecology. If you enjoyed this episode, subscribe to our show and leave us a review. To learn more about fire ecology and support our future work by becoming a member, visit fireecology.org. FIREECOLOGY.org.